Now we will be talking about polysaccharides. Polysaccharides are going to be polymers, meaning a single molecule made out of the condensation of thousands of sugar units, specifically glucose. Because of their size, polysaccharides are actually not water soluble. In table 11.4, we are going to be observing some properties of polysaccharides compared to those of mono and disaccharides. While monosaccharides and disaccharides are low molecular weight, because polysaccharides have thousands of units of sugar, their molecular weight is going to be very high. We learned already that monosaccharides and disaccharides taste sweet. Interestingly, polysaccharides are actually tasteless. When it comes to the solubility in water, many monosaccharides and disaccharides are going to be soluble in water. Polysaccharides, on the other hand, tend to be insoluble, or if any of them are actually a little bit soluble in water, they tend to be viscous. When it comes to their particle size, monosaccharides and disaccharides pass through membranes. Polysaccharides, because they are so large, they do not pass through a membrane. The first polysaccharide that we are going to learn is starch. Starch is a polymer that it consists entirely of D-glucose molecules. Starch is actually the major storage of D-glucose in plants. Starch has two polysaccharides, and they are known as, specifically, amylose and amylopectin. When we look at the top part of the images that we have on the right side of the slide, we can actually look at the structure of amylose versus amylopectin. We can observe that amylose, out of the two polysaccharides that exist in starch, they are going to, or amylose is actually having only alpha 1,4 glycosidic bonds. And these units of sugars coming together, they form kind of like a helical structure, meaning it's kind of like a spiring staircase. The other polymer that exists in starch, which is known as amylopectin, then understand that not only it has alpha 1,4 glycosidic bonds, amylopectin has alpha 1,4 and alpha 1,6 glycosidic bonds. The alpha 1,6 glycosidic bonds are going to be specifically at the branches, because one of the special features that amylopectin has is that it actually not only it has this helical form because of its alpha 1,4 glycosidic bonds, but because it has branches that are alpha 1,6 glycosidic bonds, those branches are specifically that type of bond. When it comes to chemical tests that can be utilized in order to find starch, understand that the iodine test can be utilized. Iodine is specifically molecular iodine, which is two iodine atoms connected to one another. This is the diatomic form of iodine. One of the things that it is observed is that if we look closely at figure 11.32, we can observe that iodine or molecular iodine molecules are going to be sitting on the helical portion of the alpha 1 form, I should say alpha 1 4 glycosidic bond coil that is present in amylose and amylopectin. And when it does that, it actually undergoes a color change. While iodine, we can see here in the image, is a little bit of like a yellow color. Once that iodine sits in that coil that is formed by the glucose molecules connected to each other in the alpha 1 4 arrangement, it actually changes to this blue black complex, meaning that iodine can be utilized in order to find starch in a particular sample. Next, we're going to look at glycogen. Glycogen is sometimes called animal starch. And it is called animal starch because it's the way that many animals store carbohydrates. Now, when it comes to structure, we can see that in image 11.34, we see the images that are representing the structure of glycogen versus one of the two polysaccharides in starch. Particularly glycogen, it looks very similar to amylopectin. The only difference between the two molecules is just that glycogen is more highly branched. But that being said, since glycogen looks closely to amylopectin, glycogen is also going to have those alpha 1 4 and alpha 1 6 glycosidic bonds that we observed in the molecule of amylopectin. When glycogen gets hydrolyzed, it actually helps to maintain normal blood sugar levels, and it provides energy, in particular in the muscle. And you can see in the last bullet point that it goes back and refers to the linkages between the glucose molecules that we have in glycogen. And you can see it outlined right here that these are going to be the linkages that we also observed in amylopectin. Okay. We are now going to talk about cellulose. Cellulose is the structural material in plant cells walls that provides strength and rigidity. Cellulose is a linear polymer that does not form a helix like we saw in starch. Since the glucose units in cellulose are going to be specifically beta 1 4 glycosidic bonds, one of the things that it is observed is that specifically cellulose makes an extended straight chains. Because of this organization, it is known that cellulose actually makes water insoluble fibers. As humans, we do not have the biomolecules, meaning the enzymes, to actually break apart this beta 1 4 linkages between the glucose molecules that are present in cellulose. So instead of using cellulose when we are exposed to it through our diet for energy, what humans can do with cellulose is use it as dietary fiber.